All right, I think we can get started. So welcome everyone to another edition of our Spatial Biology Masterclass. Today with me, I have James Pemberton from Standard BioTools. He's going to introduce himself in just a bit. I just want to let you know that this masterclass will take around 25 minutes, and at the end, there will be a Q&A. So you can write your questions in the chat, and we are going to read at the end. And yeah, so James, welcome. Thank you for joining today. Do you want to start with a short introduction of yourself, and then you can get started?
Yeah, sounds great. Hi, everyone. Thank you very much to Visiopharm for this opportunity to present this here to you today. My name is James Pemberton. I've been a product application scientist with Standard BioTools for a couple of years now. Recently, I've changed roles and now I'm product manager for imaging reagents. My quick background, I hold a PhD in medical biophysics from the University of Toronto, Canada, and I come from a background in microscopy, primarily like fluorescence confocal microscopy and fluorescence lifetime imaging microscopy. I've done various things, for example, like studying different cell death signaling pathways observed in cancer and neurodegeneration. But today, I'm excited to speak with you about a different and unique approach to imaging cells and tissues. So part of this talk today will discuss the instrumentation that's available from Standard BioTools for studying spatial biology. And then part two will cover Visiopharm's Phenoplex image analysis that can be utilized. So to get started, what is spatial biology? Spatial biology is a field that focuses on understanding the spatial organization of cells within their native tissue environments. So unlike traditional methods that may dissociate tissues into single cells, like single-cell transcriptomic technologies, spatial biology preserves the context of where cells are located in relation to one another within a tissue. So this approach allows for deeper comprehension of cellular behavior interactions and the microenvironment within complex tissues, such as those like tumors or normal tissue samples. So for example, I'm showing you here data collected by our research and development department. The image of human glioblastoma tumor, you can observe this on the left. And we also have a normal mouse brain tissue section on the right. What I hope you can appreciate is the vast heterogeneity of protein expression within both diseased and non-diseased tissue examples. So by revealing the spatial locations and interactions of cells, spatial biology provides those critical insights into health and disease progression and potentially to better therapeutic targets. So typical immunohistochemistry or immunofluorescence has been the starting point for a lot of spatial biology studies. But as the field rapidly progresses, we need tools that can keep up with the total amount of targets that need to be studied, the ever-increasing throughput of samples to process, and then, of course, image analysis solutions to make sense of all this data. So this is where I'd like to introduce you to Standard BioTools. We're formerly known as Fluidigm. We have a fleet of proteomic and genomic high-throughput and high-plex solutions. For example, if you're looking for ultra-high-plex protein profiling from samples with as low as 55 microliters, well, the SomaScan assay could be the solution for you. It utilizes SOMAmer technology. If you're looking for high-parameter single-cell protein analysis, our instrument for this is the Cytof XT, which excels at cytometry by time of flight technology. I'll go into a little bit more detail about this technology later in the presentation. Or finally, if you're looking for high-throughput, nanoscale, next-generation sequencing, library prep, the Biomark X9 system is a great solution for this, as well as multiple other use cases in genomic analysis. But for today's presentation, it's all about spatial biology. And today we're going to be discussing the Hyperion XTi Imaging System. This is Standard BioTools' latest platform for studying spatial biology. So some specifications about this instrument. The Hyperion XTi Imaging System, it utilizes Imaging Mass Cytometry. I'll go into the details of that technology in just a second, but just to keep it high level, this instrument is used for analyzing spatial biology of tissue sections that fit on your standard microscopy slide. It has a capacity to visualize 43 unique targets all at one time on a single tissue sample. So for myself, coming from a background of standard immunofluorescence, live cell imaging, I've shifted from looking at about 3 or 4 targets at one time to now 40 plus targets. And it's been quite the eye-opening experience going to that high plex scale. In addition to the number of parameters you can measure at once, this instrument is built for sample throughput. New this year, the Hyperion XTi now comes with an integrated slide loader. So as a user, you can stain and load 40 tissue slides, each stained with 40 plus antibodies. And you can process all of your samples within 24 hours. So this is truly remarkable scale and speed that you can work in for spatial biology. So why did Standard BioTools make this instrument? Well, I like to share a particular use case with you. This was presented by Dr. Jennifer Gorman. She's a scientific associate and lab manager at the Lunenfeld Tenenbaum Research Institute. She presented this work at the CyTOF Summit earlier this year. And if you're curious to see her full presentation, this webinar, you can find this on Standard BioTools' website. But what Jennifer Gorman sought to do was characterize a cohort of 42 patients with high-risk prostate cancer. And her team wanted to find proteomic differences between Gleason pattern 4 and Gleason pattern 5 histologies. A Gleason score, it's a grading system specific to prostate cancer. So then they wanted to assess the proteomic features and assess this in relation to the clinical measures and the outcomes, right, for the patients. So in total, there's a cohort of 42 patients, but they had 125 whole slide tissue sections that were stained with a 40 antibody panel. This very large 40 antibody panel was needed for her team to identify multiple parameters within the tumor samples. For example, identifying multiple kinds of immune cells, lymphoid and myeloid cell types, markers to study hypoxia, cell proliferation, cell death, metabolic markers, etc. So a very high parameter antibody panel on many, many samples. Dr. Gorman, they actually estimated how long it would take to acquire this data running on our previous generation imaging platform. This was just called the Hyperion, and it ran at a 200 hertz frequency. They estimated it would take almost 7 years to actually acquire all of this data utilizing that instrument, which obviously is ridiculous and not realistic. But with our latest Hyperion XTi instrument, including the new slide loader, it actually only took them about 42 days to collect and process all of this data. So again, that 125 whole slide sections, each of them being stained with a 40 antibody panel. This example is really important to highlight because if we want to answer questions within spatial biology, high parameter of targets and high throughput and processing of samples, these are some of the 2 key fundamentals that will drive biological insights. So what did Dr. Gorman's team find? Well, they are still preparing their manuscript, but when they looked at individual patient data, one of the remarkable things they discovered is the extent of heterogeneity within tissue samples from a single patient. Meaning, within a single patient, multiple Gleason scores could actually be assigned to the tumors, and it really just depended where you looked within the tumor sections. So this has significant implications, of course, for how we study prostate cancer, how it should be classified, and it also can help drive biological insights into the progression of the disease. Okay, switching back now to the actual instrument itself, the Hyperion XTi. This instrument thrives in studying high number of parameters and a high number of samples to be processed at one time. So how does this underlying technology work? This workflow is very simple, and it's very comparable to immunohistochemistry or like immunofluorescence, for example. You start by taking your antibody panel, applying this stain to your tissue samples on your microscopy slide. You insert the sample into the Hyperion XTi, collect your image data, and then proceed to image analysis. Later in the presentation, like I mentioned, I'll discuss some of the advantages to using Visiopharm's Phenoplex software for analyzing Hyperion XTi data. But how does this underlying technology work altogether? As I mentioned before, this instrument, it utilizes imaging mass cytometry. Essentially, we're generating tissue images without the use of light. In conventional immunofluorescence, you would utilize antibodies conjugated to a fluorophore and then illuminate the fluorophore to capture the tissue image. With imaging mass cytometry, or IMC for short, we utilize antibodies conjugated to unique metal isotopes, then detect these metal isotopes with time-of-flight mass spec. So within a 40 antibody panel, we're detecting 40 unique metal isotopes not commonly found in biological systems. Within the instrument, a laser beam, it ablates the tissue, sending the tissue and the antibodies to be ionized with plasma. Next, low mass ions are removed, and the remaining ions are separated by time of flight and identified. So essentially, we're identifying the metal isotopes that are present within the tissue where the laser beam was on the tissue slide itself. Then we're digitally creating the tissue image pixel by pixel. So this methodology has several key advantages in comparison to fluorescence-based methods. First, we're not using fluorescence at all. So the generated images do not display any autofluorescence signal. This is actually quite advantageous if you're working with highly autofluorescent tissue specimens. Secondly, if you are familiar with fluorescence spectra, you'll know that excitation and emission spectra, they're very wide and broad, and they do have significant overlap with multiple other fluorophores. But with IMC, imaging mass cytometry, the detection of metal isotopes using time-of-flight mass spec results in very discrete signal signatures, giving you the confidence that the signal you detect is uniquely associated with an individual target in your panel. Another advantage to IMC are the unique imaging modes available. Because we are using a laser scan across a tissue section, there are numerous ways we can sample our data. So here are 3 of the imaging modes that I'd like to introduce you to. The first one I'll introduce you to can be seen here in the middle of the screen. This is called tissue mode. Tissue mode is our whole slide imaging modality. It's fantastic for generating large tissue scans. Depending on the size of the tissue, an entire tissue section can be scanned in a matter of hours, generating data for the entire 40 antibody panel that you stain with. Next is preview mode. Just as the name implies, utilizing preview mode, as seen here on the far left, utilizing preview mode generates a preview of the whole slide image. This is extremely helpful for certain scenarios. For example, like if you imagine, if you're able to visualize the expression of all 40 markers across your entire tissue section, and if you can acquire that preview image in just a matter of minutes, usually it's on the scale of like 10 to 20 minutes, depending on the size of the tissue, that really opens the door and really demonstrates the heterogeneity that spans the entire tissue section. And that's information, that's spatial biology information you can collect within a matter of minutes. So extremely helpful for this. The resulting image that is produced can help you make qualitative assessments. So for example, let's say after collecting your preview mode image, you discover that there's high expression of a particular marker of interest in a certain area on the tissue. You can then select this region of interest and begin collecting single cell resolution data utilizing cell mode. So cell mode, seen here on the far right on the screen, is the last imaging mode that I'd like to introduce you to today. As the name implies, it's the imaging mode to be utilized if you want to get single cell resolution. And the resolution we can achieve is 0.5 to 1 micrometer. The resulting images can be processed with single cell analysis solutions. This is especially important for identifying the localization of signal. So for example, for each antibody, you may need to know, is a signal localized to the nucleus of a cell because it's detecting a transcription factor, or is the signal located within the cytoplasm of a cell, or is it located on the plasma membrane? All of this, The spatial context of where the signal is actually present within the tissue or within the cell is extremely important for answering your biological questions. Depending on the size of the region selected, this imaging mode is on the order of minutes to hours for completion. Okay. Diving in a little bit further on these imaging modes, here in this example, I want to highlight the power of combining preview mode with cell mode imaging. So here on the screen, you can see is a sample of human tonsil tissue section. And for simplicity, I'm only showing you 3 markers, CD3 in red, collagen in green, and DNA found in blue. Just showing you these 3 colors for simplicity. But as a reminder, you can visualize all 40 plus markers with preview mode. So in this example, you can visualize a CD3 T cell marker signal. And what you can see here in the image, actually, sorry, let me back up there for a second. Because preview mode does subsampling of the tissue itself, the image does appear a little bit pixelated. But the fact that you can acquire this image in about 10 minutes is where the true power comes from. It provides a wealth of qualitative information in the spatial relation of where your markers are being expressed in the tissue. So here you can clearly see lymphoid follicles and germinal centers denoted by the surrounding CD3 red signal. So the capability to image the entire tissue section within minutes enables you to capture the full picture of the heterogeneity across the whole tissue section. And this is what really informs you to make decisions on the regions of interest to go in and collect data for cell mode. So after identifying particular regions of interest, you can collect cell mode data, which is at the single cell resolution. And here in the animations, I am overlaying the cell mode image data on top of the preview mode data. And I'll flip back and forth just a little bit here so you can appreciate that the spatial information that you acquired with preview mode is fully conserved with cell mode. But of course, with cell mode, now we're getting down to the 1 micrometer resolution that we need for the single cell resolution. This workflow, it provides the fastest way to visualize whole slide heterogeneity and ensures you collect the single cell resolution data where it matters most. So instead of collecting single cell resolution data for an entire sample or tissue section, where more than half of which you probably wouldn't even use for the image analysis, you can now visualize a signal distribution across the entire tissue section and move in for those particular regions of interest to collect your single cell resolution data with cell mode. Okay, again, in this example, this is a human tonsil sample again, and here I'm just zooming in on the cell mode data so that you can visualize the single cell resolution that can be achieved with the Hyperion XTi Imaging System. The preview mode image and the cell mode images were all produced from the same slide, so there's no need for serial tissue sections in this workflow. Now I'd like to present an example of tissue mode. Imagine a scenario where you have a very large tissue section and you want to do whole slide imaging. Preview mode is excellent choice if you want to make qualitative assessments on the expression of markers across the tissue and then proceed to cell mode. But what if you actually want to capture everything across the whole slide? This is where tissue mode shines best. So you can imagine if you have no prior knowledge on the heterogeneity and the expression of markers in your tissue sample, if you solely focus on a small portion or region of your tissue, you could be missing out on such a much greater and larger big picture. So tissue mode, our whole slide imaging modality, is fantastic for generating large tissue scans. In this particular example, I'm showing you a large 5 square centimeter human colorectal tumor sample. And this sample was imaged just in a matter of a few hours. So with the ability to image the whole tissue section in just a few hours, we can identify distinct areas within this tissue section where there's different biological processes happening. So for example, in the yellow box you can see here on the right, you can visualize tumor-infiltrating lymphocytes denoted by the CD45 magenta signal. And then you can see how the CD45 signal is encountering antigen-presenting cells, represented by the HLA-DR yellow signal. Or, as you can see in the middle white box, you can identify other structures within tumor sections. In the middle box here, you can see we identified a tertiary lymphoid structure. So tissue mode generates images at a resolution of about 5 micrometers. Quantitative analysis can be done utilizing pixel clustering analysis or by using Visiopharm's tissue segmentation app, which I'll dive into a little bit further on here. Okay, first, I'd like to explore a little bit now into the data analysis that occurs with the image data that comes from Hyperion XTi. You can utilize a variety of image analysis solutions. So for example, ImageJ, Halo, Cell Profiler, and of course you can use Visiopharm's Multiplexed software. Two things to note. The first being that with the purchase of a Hyperion XTi instrument, you do receive a 1-year license for Visiopharm Multiplexed. And then secondly, if you do want to take advantage of Standard BioTools IMC services, our Discovery Lab Imaging Mass Cytometry services, they'll process your data utilizing Visiopharm's Multiplexed software. Okay, to start, the file that's exported from the Hyperion XTi is called an MCD file. You can extract the OEM TIFF files for each marker and then proceed to analysis. Alternatively, Visiopharm makes it easy, and you can directly open MCD files within Visiopharm. In this first view of the software, you can visualize all the channels, assign pseudocolors, and adjust image brightness and contrast. You can also create groups. That's what you can see here on the far right that I started doing. Any group, really, it depends on what it is that you are studying. In this example on the far right, you can see I've created a group called just T cells. And this group is defined as the expression of CD45, CD4, and CD8, all of which were antibodies included in my panel. Another group potentially you could do would be tumor cells or epithelial cells. Maybe you're looking to identify cell types associated with blood vessels or infiltrating immune cells. It really depends on the biological question you want to answer. But the main take home is you can create these groups, and the groups are defined as the markers that you assigned from your panel. So when processing your cell mode data, as a reminder, this is your single cell resolution data, you need to first do cell segmentation. To answer your biological question, it's important to distinguish the location of signal. Is it within the nucleus, the cell body, across the entire cell? This is all information you can extract using Visiopharm Multiplexed. So to do this, you first start off by using the cell detection app. It utilizes AI to automatically identify individual nuclei and cells, and it can be used for batch analysis for all your image data. We find the segmentation app actually quite accurate when analyzing our data. Multiplexed also has some other useful tools. So for example, there's measurement tool. Oh, I think I went ahead by one slide. A few other useful tools that you can utilize once you segment your cells as well. Measurement tool, if you want to quickly determine the size of structures within your image data. An annotation tool, if you want to create a note based on the visual observations. And a few other tools that are quite useful, especially if you need to revisit your data later on. Something that usually happens. The next step in the image analysis using Visiopharm is to apply a thresholding or gating. So you need to determine what signal should define objects of interest in your data. So in this particular example I am showing you, I'm creating a gate to determine what CD3 expression should define a CD3 positive T cell. And then in real time, you can then visualize, so the gating here is on the left, and then in real time, you can visualize what cells fall within your gate on the right. So on the right, you can see this is the cell mode data. You can see the segmentation mask, and you can see these individual yellow dots represent the cells that fall into my defined threshold gate that I applied. Okay. What else can I tell you for this slide? I think that's it for this one. Now that we've done the cell segmentation, we've done our thresholding and gating, now we're at the point in the analysis where you can actually start to answer some of your spatial biology questions. So, for example, utilizing the co-occurrence matrix, this can tell you the occurrence of 2 or more markers within the image data. So in this example, we created a co-occurrence matrix for 7 markers. And this co-occurrence matrix is telling us right now that about 95% of the CD20 positive cells are also CD45 positive. And this, of course, makes biological sense, knowing that CD45 is a panleukocyte marker and CD20 is a B cell marker. So we, of course, expect a very high co-occurrence to occur. Visiopharm also has multiple neighborhood analysis tools available. These are extremely important in the study of spatial biology, as we want to understand the spatial context within the tissue sample. So this first tool is called neighbor counts. Essentially, if you imagine you have a cell located within your tissue, and let's call it a CD3 positive T cell, the question you want to ask is, what kind of neighbors or other cell phenotypes are within a certain distance of this kind of T cell? You can adjust the distance for proximity. 1 to 30 micrometers is the default set in the software, but it really just depends on your research question. If you're asking what phenotypes are in close proximity to your target phenotype, it then provides you a mean standard deviation count and tells you what these close proximity cell types are. Now, in relation to this, another very important spatial biology, spatial analysis tool is the nearest neighbor tool that Visiopharm has. So neighbor counts helps you identify the neighbors or the cell phenotypes, what they are. Nearest neighbor tells you how close are the cells together, providing you a mean distance for the different cell phenotypes. So continue our example here. You can use neighbor counts to identify the cell phenotypes that are in close proximity to the CD3 positive T cells. Then you can use nearest neighbor to tell you how close are these cell phenotypes to the CD3 positive T cells within the tissue. This can help you identify cells that may have functionally direct interactions with each other, cells that may be part of a signaling network, and for example, like the proximity of certain immune cell types that can really help inform you of the immune response that may be occurring within the tissue sample. Again, it comes down to what is your biological question. Utilizing both neighbor counts and nearest neighbor tools, it's essential to answering a lot of your spatial biology questions. Okay, and then finally here in the analysis, what I've shared with you so far is a quick overview of the image analysis that can be utilized for cell mode data at that single cell resolution. But Visiopharm Phenoplex, it does also have some solutions available for tissue mode data. So the most useful of this would be the tissue segmentation app that they have. It enables you to identify specific regions within the tissue based on marker assignments. So for example, you can create image classes named like tumor, non-tumor, and you can train the app to recognize parts of the tissue as tumor-based based on the expression of the certain markers that you assigned. And then you can use this AI to do batch tissue segmentation on all your tissue mode data. So extremely useful for segmenting and identifying different regions on your tissue mode data. Okay, wrapping up this presentation here for today, I know I've thrown a lot of information at you here. We've reviewed the Hyperion XTi Imaging System. We learned how imaging mass cytometry works. We explored the different imaging modes available with the Hyperion XTi. And we got a taste of the image analysis solutions offered with Visiopharm's Phenoplex software. All of this, I understand, it can be a little overwhelming, especially to users who may be new to multiplex spatial biology. Getting started may seem like a daunting task. So, for example, starting at the very beginning, how do you design a 4D antibody panel? Well, at Standard BioTools, we do offer pre-configured antibody panels. We now have over 25 IMC panels currently in our catalog, each relating to different research applications. So, for example, human or mouse immuno-oncology, neurodegeneration and phenotyping, T-cell exhaustion, tissue architecture, etc. Starting with one of our pre-configured panels makes the process of designing your high parameter antibody panel that much easier. Plus, you can also bring your own antibodies. If there's a target not available in our catalog, you can easily add it to our IMC panel. You can easily add it to your IMC panel so long as it has some previous history of working with immunohistochemistry or immunofluorescence, the clone will most likely work then with imaging mass cytometry. The last thing I just want to mention here today is that if you are looking to get started with imaging mass cytometry, a great place to start is with Standard BioTools services. You can ship your FFPE or fresh frozen slides, and you let our scientists do the work for you. Our scientists, they'll do the antigen retrieval, the antibody staining, the data acquisition, and the analysis for you. So you can use our services to gain pilot data. You can gain spatial proteomic data to complete that manuscript that's currently in the works, or you can utilize it to gain data for grant applications. It really depends on your particular use case. But our scientists, they provide the data analysis for you utilizing Visiopharm's Phenoplex software, as I just showed you here today. So with that, thank you all very much for your time and your attention today. I'd be more than happy to answer any questions that you may have.
Wow, thank you so much. Let's get started with the Q&A. First question, you talked a lot about Phenoplex. I'm just curious, what is, if you had to choose one thing about Phenoplex that you love the most, combined with Hyperion images, what is it?
Good question. I think one of the very useful tools is, or abilities, is the fact that you can just directly take the raw data that comes off the instrument, the Hyperion XTi. This is the MCD file, and the fact that you can open it and start processing your data using Visiopharm Phenoplex. So you don't have to go through the trouble of trying to convert all of the image data to the OEM TIFFs first, trying to export, save them somewhere, and then try to re-import them into Visiopharm. That's one really nice thing I appreciate with the software.
Cool, thanks for sharing. So I can see that we have some questions already. So Hasmus, he wrote a question, I think at the beginning of your presentation when you were presenting the case study, I think. So he asked, is that 24 hours total acquisition time for the entire whole slide, for all 40 slides in the slide loader at 1 micrometer resolution?
Yep, that's referring to, I think, the one slide where I introduced the Hyperion XTi and the slide loader as well. Yeah, so we advertise it as you can do 40 slides, 40 antibodies, and you can accomplish this in 24 hours. In that particular example, each slide, you're selecting, I believe, 3 regions of interest to collect cell mode data. So in that example, that's not collecting data across the entire tissue section. That's 3 individual regions per tissue slide. If you want to get a general sense of how long it takes to acquire data with the Hyperion XTi, it really comes down to area. So other technologies like cyclic immunofluorescence, the amount of time to acquire all of your data is dependent on the size of your antibody panel. For us, the determining factor for how long it takes to acquire data is the area of the tissue itself. So a larger area takes a little bit more time, smaller area, smaller time.
Cool, thank you. So let me see, question from Nareg. Can you quantify expression levels, and have anybody tried staining for post-translational modifications like phosphorylation? Have you tried to reconstruct a whole tumor 3D?
Oh, good question. On the 3D front, I know of 2 publications. The titles of the publications are escaping me. There's 2 publications available where 2 independent groups looked at doing 3D reconstruction using imaging mass cytometry. Now, one approach could be you could do multiple serial sections of a tumor. So if you have multiple serial sections, each section you would stain with the same antibody panel. You would then process each one individually, and then you can do digital reconstruction to create the 3D image. Another approach is actually taking a much thicker tissue section, staining it with your antibody panel, and then doing subsequent scans on that tissue. So for imaging mass cytometry, how the technology works, it's a laser that goes across the tissue section and ablates a very small layer of the tissue. So essentially what you're doing is you're continuously ablating a smaller and smaller layer of the tissue. You collect that IMC data, and then you can reconstruct that into 3D. Sorry, I think that was part 2 of the question. Was there a part 1?
Can you see the chat? Can you quantify expression levels and have...
Oh, can you quantify expression levels? Really good question. Depends what you mean by quantify. If you want absolute quantification, that currently does not exist. But if you want to, with the Hyperion XTi, the dynamic range of signal you can detect within a single sample, you can detect very low signal, and you can also detect very high signal. The dynamic range for epitope expression is quite large. So this is quite useful in studies where you might want to stratify samples based on the expression of a certain marker, and you want to be able to capture that entire range. That you can do, what's it called? Qualitative expression levels.
Great. So next question from Ali. How many parameters do you recommend to get an optimal segmentation? Is DAPI only enough to work on IMC data?
Yeah, great question as well. So how many parameters do you recommend? We do offer a cell segmentation kit. This kit is 3 antibodies that bind to unique targets on cellular membranes. So if you use the segmentation kit, 3 markers to identify different regions of the membrane, and then you use not DAPI, because DAPI you would use to identify nuclei in fluorescence technologies. For us, you're going to use iridium metal, so iridium 194, I believe. So you'll have your 1 DNA channel to identify the nuclei, and then you'll have 3 markers to help identify the cell membrane.
Thank you. Next question from Mass. Since IMC works with rare metals, could you use non-antibody conjugated metals for IMC? For example, loading microvesicles with specific rare metals and then use IMC to detect where they end up in a tissue slide?
That's a great question. Yes, you can, technically. I'd have to go look at some literature for this, but what you are detecting is the metal isotopes. So whether or not these metal isotopes are conjugated to the antibody, or in another application that I've personally done myself, if the metal isotope is conjugated to an oligonucleotide for detecting different transcripts in the tissue, that also works. Or in your particular example there, yeah, if that specific metal isotope is somewhere found within the tissue, we should be able to detect it with imaging mass cytometry.
Cool. Shannon is asking, is there any information on what markers are used in the cell segmentation kit?
That I would have to go back. I think that's proprietary that we have on the segmentation kit itself. It's 3 unique cell membrane protein markers. Stains usually most different cell types or tissue types that you would encounter.
Okay. And Sonia asked, if someone is designing their antibody panel to be used for imaging mass cytometry, where do they get the metal tagged antibodies? What if they cannot find a particular clone that they want?
Yep, great question. So there's a few different options that you can go with. Like I mentioned, if you have an antibody or clone that's worked well with immunohistochemistry or immunofluorescence, it'll most likely work on imaging mass cytometry, like the Hyperion XTi platform. You can start by looking at our catalog of pre-conjugated clones and clones with different metal tags to build your panel. If you can't find a clone of interest and you have one yourself, you could either conjugate the antibody yourself with the metal tag. We sell a conjugation labeling kit for this. Or you could actually send in the antibody to us, and we provide custom conjugation services. So we'd be able to put the metal tag on for you, send it back, and then you could proceed with your imaging mass cytometry experiment.
Cool. Thank you. I believe that was the last question. So if anybody has additional questions at some point, we are going to send an email with the recording, and yeah, you can always get back to us or James. James, thank you so much for the masterclass. It was really great having you here, and everybody that engaged. It's always so nice to see you here. We have a last minute question actually from Nareg. Would that conjugation service be available for other commercially acquired labs or ABS?
For commercially acquired antibodies? Yeah. Any antibody that you can buy from a commercial vendor, one that you might have sourced from your own lab, you just send it to us and we conjugate it to you as a service and we ship it back to you. Or if you wanted to go through our Discovery Lab services so that you could access the Hyperion XTi through our service lab, you could still send in the antibody. We'll do the custom conjugation and then we'll apply it to your sample when we run the experiments.
Perfect. Thank you once again. And yeah, thanks everyone for joining. Thank you. So yeah, have a great day, evening, morning, everyone, depending where you are. And don't forget to follow us on LinkedIn as well. We are always promoting our masterclass series there, so you will know when there's a new one, when you can rewatch the recordings. So
fantastic. Thank you very much.
Thank you.
Bye, everyone.
Bye.